Hi everyone. For those of you who don't know me, my name's Christopher Cornwall. I am a researcher at Victoria University Te Waka here in New Zealand, and for the last 13 years my research has assessed how coralline algae and other seaweeds and corals as well will respond to the impacts of ocean acidification. Today's presentation is going to discuss a lot of this recent work. So maybe 13 years ago, I began working on this with Katrina Hurd, and recently did a postdoc in Malcolm McCulloch's group over in Australia, and now for the last 3 years I've had my own research group here in New Zealand. So a lot of this work is going to involve a lot of my collaborators and co-authors. So today's presentation is taking together the best parts of maybe 4 different papers and putting them together. So Steve Camo is a co-author in all of that, and the majority of this talk is going to be revolving around some work that both myself, Steve Camo, Ben Harvey and Lucia Porzio all co-led. And so I'd like to really acknowledge those guys right now, as well as others involved in this work. So for those of you who don't know what coralline algae are and why they're important, what we can see here in the slide are all coralline algae. And the coralline algae are basically a calcifying red seaweed. They can form a veneer over rocky reefs. They can also form their own thick contributions of calcareous calcium carbonate in some temperate zones and also in the tropics. Not only do they perform this important cementing role, they can also create their own reefs in many places in the tropics. As well as that, they emit chemical cues into the seawater that enable many marine invertebrate larvae to return to these reefs in the tropics and in temperate zones. And these include ecologically and economically and culturally important species such as paua or blackfoot abalone here in New Zealand, but also things like coral larvae. So without coralline algae, the functioning of these reefs is going to be heavily perturbed. And an example of this, the massive ecological importance of coralline algae, is pictured here at Montgomery Reef. So Montgomery Reef is actually Australia's largest inshore reef. It's 380 square kilometres of coralline algae. What you can see here, all the brown stuff, is essentially coralline algae. What we see is white patches on the sides of that reef, which are bleached coral communities during this massive 2016 El Nino event. Now this photo was taken by Steve Camo from a plane. If you were to zoom out, you'd see me down below on the reef itself. This reef is home to an incredible array of biodiversity, including crocodiles, whales, sharks, and a lot of coral communities. Now, for example, the size of this reef, those gouges that you can see up the middle, I've seen in a been in a boat when it's driven up them into the reef, seen footage of whales, etc. swimming up them. So these are massive, massive areas of coralline algal reef. You stand on one end of the reef and you can see coralline algae in all directions. Now, contrary to some opinions, this reef and others like it are actually comprised of multiple species of coralline algae. So not just the Pyroliphan uncoides that forms on traditional coral reef crests. This is comprised of hydrolithons, lithophyllums, etc. Now the problem is, what we can see here during this coral bleaching event is that increased temperatures are badly impacting these coral communities. However, the coralline algae themselves are not actually bleaching during this event, and in other locations nearby they respond similarly. So coralline algae are a bit more resistant to the effects of warming than corals are, for example. However, what we do see at these places is strong impacts of ocean acidification when we take these coralline algae back to the lab. Indeed, this is something that comes through in the literature quite strongly. So around the 2008 time period, there were an array of really seminal papers that were published that assessed the impacts of ocean acidification on coralline algae. One of which, for those few people in the audience who haven't read this paper, is that paper by Hall, Spencer et al. 2008. What they find at the top here, we have different stations. So South 1 having control seawater pH. This is in Ischia, Italy, where there's a natural CO2 venting site. Station 2, the seawater is more similar to that we expect at the end of the century due to ocean acidification, although with a bit more pH variability in there. What we see is a switch from calcareous communities, high calcareous algal cover, to low calcareous algal cover as we move towards this venting site. And similarly, we see an increase in the non-calcareous algal cover at these locations. So this study and others around that same time period published really strong results showing that ocean acidification will have severe negative ramifications for coralline algae, and included in that is the study by Koffner et al. 2008, where they show that coralline algal recruitment is strongly perturbed by the effects of acidification. Other papers like Martin and Gattuso, Anthony et al. 2008, these papers were all really similarly seminal, so apologies for not mentioning those. But one of the motivations that myself and others that I'm working with had was understanding whether or not these effects would hold true. So hundreds of research papers later, do similar effects manifest on coralline algae? So for those of you who are aware of the fish literature, there's been a discussion of this decline effect. Does this occur for coralline algae? What we wanted to do was not only discover that, but also understand how coralline algal physiology is impacted by the effects of ocean acidification in the systematic review / meta-analysis approach. We found 298 papers that had investigated the possible impacts of ocean acidification. So not only did we extract data from these papers, we read them and summarized their results in this analysis. We found what we call 392 response curves that assess the impacts of OA on coralline algae. What I mean by response curves is that we have a control treatment and at least one other OA treatment in this paper. From that, it was obvious the effects of calcification, the effects on calcification or growth in the laboratory, and on field recruitment and cover were most commonly assessed. We then did a quantitative meta-analysis on the responses of calcification or growth to different pH treatments in the lab. By responses, what I mean here is that 543 different responses are responses to a specific treatment. So these include control treatments as well. And what we find in situ, and thanks to Ben for putting these figures together, what we find in situ is strong negative effects on coralline algal cover in A and on recruitment. So as we intensify acidification, we get much lower cover and much lower recruitment. And what we see in the laboratory is we decrease pH, we get a greater effect size. So these points all represent the hedges G. And so what that's demonstrating is the response relative to the control. So anything under the 0 is a negative response. What we see in adults, apart from a few outliers, is strong negative responses to intensifying acidification. What we see in juveniles is similar, but we have a much smaller range and a much smaller sample size. What we also did was we explored all the patterns of potential covariating factors. So think about the ocean basin from which the study was conducted, things like order, family of the coralline algae, the duration of experiments, light intensities used, temperature used, any other covariating factors that we could assess. What we found was there were so many covariating factors that all masked each other's effects that it was very difficult to determine patterns. What we did find was significant impacts of decreasing seawater pH. So as we intensify acidification, we get much stronger negative responses of calcification and growth to decreasing seawater pH. What we also found was a few interesting taxa-specific responses. So one particular subfamily or family responded positively to the effects of ocean acidification across almost all studies assessed. And also some of the sporolithins we found had more robust responses, particularly in the juveniles, although that was not statistically significant in our analysis. What we then did was we put all what we call the qualitative assessment together. So this is essentially the systematic review, but numerical. What we found in earlier studies was strong impacts on recruitment, cover, calcification. These come through across the board, so no such decline effect is detected here. What we can see is in red are negative responses, and in pink are parabolic responses, so they respond initially positively, then negatively after seawater pH declines. In black, we have a different sort of category of negative responses. So this is not only do they respond negatively, but really strongly. So for recruitment, it means there was zero recruitment. For cover, there was zero cover. For calcification, it means that they were dissolving rather than calcifying under OA. And the grey treatment's been no measurable effect. So this is occasions where there was not a statistically significant effect. And in blue is a positive effect. So we look at recruitment, strong negative impacts on recruitment, not surprising based on the figure I showed previously, and the same with coralline algal cover in the field and in mesocosm experiments. Calcification rates strongly negatively affected. Now, calcifying fluid pH, this is something that might not be that known by the general audience. So the calcifying fluid in coralline algae and in corals as well is more of instantaneous pockets of precipitation of calcium carbonate rather than an actual fluid per se. So we can't get a probe into this fluid, but we know using geochemical proxies that the pH in this fluid is elevated, and I'll go into that in a minute. What we see is below these consistent responses is some other highly measured responses to acidification. What we can see here are what I would classify as inconsistent responses. So mineralogy, did they increase or decrease their magnesium content? If it decreased, it's in red here. What we see is a few studies earlier on that found such a thing, and some of my research has as well, but not all species do this. So not all species decrease their magnesium content. Some of them actually increase, but the majority of them don't change at all. Photosynthetic efficiency and pigment concentrations can sort of be grouped into the same sort of response that we'd predict. So if they're stressed, their photosynthetic efficiency and their pigment concentrations are likely going to decrease. If they're bleaching, think about how corals respond to marine heat waves. If they're bleaching like a coral responds to a marine heat wave, then this will be a consistent negative impact. What we see here is actually some positive impacts and generally inconsistent, so no measurable effect. Now, we do see some negative effects, so either some species are responding negatively or there isn't a true impact here. What we also see when we dig further into some of these studies is ecologically non-relevant declines in photosynthetic efficiency. You're talking about 0.02 units, so they're still healthy, but their photosynthetic efficiency has dropped somewhat. Remember that they're in an artificial situation, and most times handling stress, etc. I've observed personally that even without OA, if you increase handling stress to coralline algae, this photosynthetic efficiency will decrease. Photosynthetic rates, again, we would expect to see a consistent negative effect here if OA was a physiological stressor on their photosynthetic apparatus. However, what we see is, again, inconsistent responses, and we see some positive responses. So obviously some species, or in some experiments, they are responding negatively, others they respond parabolically, and then in a few that we see what's probably alleviation of any dissolved inorganic carbon limitation. So coralline algae have a range of different carbon concentrating mechanisms, or CCMs, that allow them to actively pump bicarbonate into the cell. And this doesn't mean that they're not going to be DIC limited though. And that's probably what's occurring in some of these occasions, is alleviation of DIC limitation. So to answer the question of do the older studies hold, most of the findings that coralline algae are going to be badly impacted by acidification do indeed hold. Although some of the physiological measurements that underlie these responses aren't responding as negatively as some of the more ecological responses, such as recruitment, cover, and calcification. Now, there's been some talk in social media particularly about whether OA is actually important or not. And that's where this concept of the dominant driver comes in, or the primary driver. What we assessed here was when OA is assessed next to another driver, how often is OA having a larger impact than the other driver, or how often is that other driver having a larger impact? So this is the concept of this primary driver. There are some discussions that OA is sort of similar to microplastics and that it will have negligible or no impacts on marine systems. We think this is not true based on the results we're showing here, particularly for coralline algae. So some drivers could be more important than others for different taxa. For example, for corals, temperature is going to be most likely the primary driver, but for coralline algae, what we see consistently, if we ignore seasonality here, is that OA is more often than not the primary driver than any other driver, both in terms of the frequency in which it was measured and the proportion, so the proportion in brackets here. Interestingly, seasonality is a really strong driver here, and that's something that probably shouldn't be ignored in future research. But things like temperature are not, and light. Light is really surprising for those of us who are seaweed biologists or physiologists. We know that light primarily drives their physiology, so the fact that OA has a stronger impact on coralline algae is saying something here. Interaction types. Now, for those of you who haven't read the Crane et al. Paper and others before it, what we're looking at here is synergistic or antagonistic effects. The proportion of times these have been uncovered are in brackets. Synergistic mean we add both the drivers together, they have a larger impact. Antagonistic mean we add two drivers together, they have a smaller impact than they do individually, or they counteract each other. What we also have is an additional category. So additive is the response that we'd predict based on the sole responses to singular drivers by themselves. So what we mean by no measurable effect of one or more variables in white here is a different category that we decided to show for demonstration purposes, but can be categorized as additive. This was interesting to us. The reason why is because, now I'll explain what this is first. Firstly, we would, based on previous literature, assume that synergistic and antagonistic effects would be in around the 50% proportion. That's not what's occurring. Often what's happening here is that OA has such a strong impact on coralline algae that the other driver simply has no measurable impact when compared to that driver. So, for example, if you were to run an experiment assessing how ocean acidification and warming impact coralline algal physiology, and they respond negatively to OA, but don't respond at all to temperature, then when you add OA and warming together, they respond exactly in the same way they did to OA. That's what we mean by this white category here. That was really surprising. But what was surprising to us was that some of these factors have something in common that come through as having synergistic and antagonistic effects. And that's that light, presence or absence of epiphytes, and water velocity all have high proportions here. These are factors that can impact pH at the scale which really matters for coralline algae, which is within what we call the diffusion boundary layer, right? So in this diffusion boundary layer, it's a layer of seawater where the movement of dissolved substances is primarily via molecular diffusion. So during the day when they photosynthesize, they take up CO2, pH is quite high in this layer, and there are a range of other physiological pumping that go on that change pH there, but we won't go into that for today. Now, the problem with coralline algal physiology is it's extremely complex, but it's clear from this analysis and from previous studies that pH in the diffusion boundary layer really can dictate responses. Now, this diffusion boundary layer pH is not primarily what's controlling calcification, but rather, we believe, it's impacting pH inside the organism, so in this calcifying fluid pH. Now, there are some people out there that consider the diffusion boundary layer pH as the same thing as what we call pH-CF. This is not true. So we have made measurements of this pH in the diffusion boundary layer against pH in the CF, and pH-CF is also always much higher. And what's more complex is that here we have a figure from a paper in 2017 where we tried to, in our own way, put together the different taxa, coralline algal taxa, and how they calcify. In hindsight, you can't really do that. Different taxa are calcifying in different ways. So not only do they have different ways they lay down the calcium carbonate crystals, but they also have potentially got different layers of cells between the seawater and where they're calcifying. So they could be calcifying several cell layers deep, so not in the diffusion boundary layer. Even those that are calcifying on that initial surface cell are calcifying internally, we believe, in their cell wall. But that cell wall fluid, or pocket, of the site of precipitation is heavily influenced by seawater pH, but also metabolic pumping. And this is really where the field needs to move to understand what sort of movement of ions, etc., is occurring here. So for corals, we have been able to, as a community, not myself, we have been able to calibrate many geochemical proxies. So the way that we measure pH in the calcifying fluid is that we know that boron isotopes track the pH of the fluid in which the calcium carbonate was laid down. This has been calibrated for corals extensively and for coralline algae now. What we don't know for coralline algae is what dissolved inorganic carbon or the saturation state of that fluid is. For corals, these inorganic seed experiments where they take aragonite seeds, they grow them at different seawater carbonate conditions, and so different DIC concentrations, different pH values, saturation states, temperatures, etc. They can tease out how that fluid impacts the geochemical proxies, and in this case, boron-calcium ratios, which dictate DIC in the fluid, and Raman spectroscopy derived values which dictate saturation state, or proxies for saturation state, I should say. But for coralline algae, we have the same proxies, but we can't numerically determine what they mean. And that's really the next step for our field. So for example, for corals and some coralline algae, we can put them into these different boxes. So why do coralline algae and corals respond negatively to acidification? The majority of species which respond negatively have calcifying fluid pH declining under OA. So as external seawater pH declines, internal pH declines. That DBL pH can offset it somewhat for both corals and coralline algae. What's also occurring for corals is that DIC is increasing, and the same proxy in coralline algae responds in the same way generally. So we can place coralline algae and corals into this box. But for unaffected species, we know that many species have pH-CF homeostasis. So as pH declines externally, the pH is constant, calcification rates are constant. We can put a couple coralline algae into this box and quite a few corals. However, we can't determine for coralline algae the next thing, which is for some corals, they can actually compensate for declining pH in the calcifying fluid by taking up more calcium. And that's the next step, is really to calibrate this. Skip through the slide. What also is interesting, and just wrapping up here because we've only got a few more minutes to go, is that the responses of adult coralline algae, which is primarily what I was talking about in the previous slide, doesn't always carry through for juveniles. But what is interesting is that we know in some of the few multi-generational experiments that coralline algal juveniles can increase their tolerance to the effects of acidification if grown for multiple generations under OA. So what we have here in generation 2 is essentially recruitment. How is recruitment impacted by ocean acidification? So the control being the white bar, OA being the red bar for that generation 2. Now, we need to move beyond generation 2 to go beyond measuring just the effects on recruitment. So what we see after subsequent generations, and by the time we get to generation 6, there's no effect of OA. So we see increasing tolerance over multiple generations of exposure in that OA treatment. And then in generation 7, what we did was a reciprocal transplant experiment. In this particular study, what we did was we took half the individuals from the OA treatment, placed them into the controls, and vice versa. And then kept half of them in their own treatments. What we see is individuals that are placed into this novel OA treatment from the controls respond much more poorly than individuals that have been grown for 6 generations previously under OA. And if you're interested in these results, check out Cornwall et al. 2020 from last year, and also this year Moore et al. 2001 in Proceedings B, which is showing that reproductive output is also changing in some of these individuals. So we know that coralline algae can gain tolerance, or at least this particular species, which is a type of hydrolyphin, can gain tolerance over multiple generations. However, the geochemical proxies that I just spent 10 minutes talking about don't track this change in tolerance. And actually, the pH in the calcifying fluid is still decreasing under robust species, so they don't fall into that same category that, say, for example, the Porides corals do. What's interesting is this is the full width half maximum using Raman spectroscopy. This is a proxy for saturation state in corals in their calcifying fluid. So for the juveniles, we're seeing here that actually when they're calcifying faster, that their saturation state is lower, and what we believe is occurring is that only individuals that can continue to grow when the saturation state is low do so. And what we need to do now is actually be able to numerically determine what this value is to determine pumping of calcium and other such things. So we know for corals that this also occurs in select species. When they grow rapidly, they take up calcium and deplete the saturation state in that CF. And that's probably what's occurring in this study as well. Now, just to go from the complete physiological scale back to ecology. So why is ocean acidification important, and how will it manifest in the real world? This is a study from PNAS this year, Cornwall et al. 2021. What we looked at was firstly present day reef growth. When we look at the contribution of corals and coralline algae to reef growth, and then we modelled the effects of ocean acidification, warming and marine heat waves on these reefs. They have different geographical locations in the world, so these stresses will manifest at different times and in different magnitudes, and they have different contributions of corals, coralline algae, different bioroders, etc. What we see under RCP 2.6 is, not surprisingly, not too much of a bad response, but what we do find is a 77% reduction overall in coral reef growth globally. Under RCP 8.5, what we see is an intense destruction of coral reefs. So marine heat waves have essentially killed off most corals under this scenario. What we see is almost every reef in the world is eroding. We do see a couple of spots where the reefs are still growing, and these reefs all have one thing in common. These reefs have presently got a high contribution of coralline algae at these sites. However, even at these sites, that ocean warming is not having strong effects on the coralline algae, but acidification is having intensifying impacts. So if we look at the table in this paper, and have a read of this paper if you're interested, what we see is stronger effects of ocean acidification on coralline algae than on corals. We've seen 27% reductions by the end of the century in coralline algal net calcification here, which isn't surprising based on the results that I showed previously. Now, this is important because there are some reefs in the world that are completely comprised currently of coralline algae. We don't know whether these types of reefs will increase in frequency in the future. These are reefs that generally are really thermally variable, so the corals can't really live too well on these reefs. So really high temperatures, up to 40 degrees, for example, at Talon Island, pictured in the top left. These reefs are comprised of not only reef crest forming porolithon, but also a lot of hydrolyphin, lithophyllum, neogoniolithins, etc. We need to better understand how these reefs will respond in the future to acidification by exploring the taxonomic variability in their responses. The problem with our meta-analysis is that for some families and orders, etc., there was such few data that we couldn't properly explore whether some of these species were more resistant or not. And we need to pair these examinations with full assessments of physiological metrics, including more of these geochemical proxies. And so that's really where I think the field needs to move now, just to quickly wrap up, is to improve our model of calcification by understanding both the using old school techniques and the new age geochemical proxies. We need to calibrate these high magnesium calcite seed experiments. So we don't have these same calibrations to understand what these proxies mean numerically for coralline algae, because the high magnesium calcite, or the high magnesium content in the skeletons, we believe, is perturbing some of these results. In the seed experiments, we also need to get a better prediction of reef scale coralline algal calcification and better understand coralline algal contributions under climate change because of this, and further explore these eco-evolutionary mechanisms of changing tolerance and other species, not just the hydrolyphin that's important in the Kimberley area forming reefs, but also these reef crest forming pyrolithins that form in more traditional coral reef settings. And that's all. Thank you very much.